Hello, my name's Jeremiah J. Pusa, and I've decided to do a reading challenge for equipping myself with my PhD in immunology and vaccine. Therefore, if at any time you're interested with this, you'll join me and you can easily follow me because currently I'll be reading the book called Cellular and Molecular Immunology, written by Abdul Abbas Andrew Lichman together with the Shiv Pillai. This is 10th edition. The publisher is Elsevier, and we'll be going through the topics from the start to then to an end. And as per today, my discussion really will be the first part of the immunology, whereby we'll be discussing as whole and I'll be reading as challenge. So let us start our discussion with the first chapter in the book of immunology. As I said earlier, so we'll be discussing from the book of cellular and molecular immunology. The first chapter will be our discussion. Let us start. Chapter 1, Properties and Overview of Immune Responses. Here, we'll start about innate and adaptive immunity, innate immunity, adaptive immunity, whereby we're going to look for cardinal features of adaptive immunity response. We'll look of overview of humoral and cell-mediated immunity, initiation and development of adaptive immune responses, humoral immunity, together with cell-mediated immunity. Thereafter, we'll look on summary. So the term immunity is derived from the Latin word immunitas, which refer to the protection from legal prosecution offered to Roman senators during their tenure in office. Historically, immunity meant protection from the disease and more specifically infectious disease. The cell and molecules responsible for immunity constitute the immune system, and their collective and coordinated response to the introduction of foreign substances called immune response. The physiologic function of immune system is defense against infectious microbes. However, even non-infectious foreign substance and product of our own damaged and malignant tumor cell can elicit immune response. Furthermore, mechanisms that normally protect the individual from infections and the elimination of foreign substance also are capable of causing tissue injury and disease in some situations. In some situations, even self-molecule can elicit immune response, so-called autoimmune response. Therefore, a more inclusive definition of immune response is a reaction to microbes and to a molecule that are recognized as foreign or abnormal, regardless of physiological or pathological consequences of such a reaction. Immunology is the study of immune response in this broader sense and of the cellular and molecular events that occur after an organism encounters microbes or other foreign macromolecules. Historians often credit Thucydides in the 5th century BC in Athens as having the first mentioned immunity to an infection that he called plague, but that was probably not the bubonic plague that we recognize today. The concept of protective immunity may have existed long before, as suggested by ancient Chinese custom of making children resistant to smallpox by having them inhale a powder made from the skin lesion of the patient recovering from disease. Immunology, in its modern form, is an experimental science in which explanation of immunological phenomena are based on experimental observation and the conclusion drawn from them. The development of immunology as an experimental discipline has depended on our ability to manipulate the function of the immune system under controlled conditions. Historically, the first clear example of this manipulation and one that remained among the most dramatic ever recorded was Edward Jenner's successful vaccination against smallpox. Jenner, an English physician, was aware of an observation in rural England that milkmaids who had recovered from cowpox did not contract more serious smallpox. On the basis of this observation, he injected the material from a cowpox pustules into an arm of an 8-year-old boy. When this boy was later intentionally inoculated with smallpox, the disease did not develop. Jenner's landmark treatise on vaccination, the Latin Vaccinus, of or from cow, was published in 1798. The principle of infectious disease and vaccination were firmly established by the work of Louis Pasteur and Robert Koch 100 years later. These advances led to a widespread acceptance of the method for inducing immunity, and vaccination remained the most effective strategy for preventing infectious disease. An eloquent testament to the importance of immunology was an announcement of the World Health Organization in 1980s that smallpox was the first disease that had been eradicated worldwide by a program of vaccination. The significance of the immune system has been dramatically and tragically highlighted by the AIDS, acquired immunodeficiency syndrome, epidemic caused by HIV, human immunodeficiency virus, that started in 1980s, and the COVID pandemic caused by coronavirus SARS-CoV-2 that started in 2019. Both have caused severe morbidity and many deaths, and have a devastating impact on society. Development of effective vaccine for both diseases is a high priority. Effectiveness of vaccine of some common infection have been shown in a number of the ways. For example, in year 1921, diphtheria had 206,939 cases, but as of 2018, 1 case remained. Measles in 1941 had 894,134 cases, but in year 2018, 375 cases remained. Mumps in 1968 had 152,209 cases, but as 2018, 2,515 cases remained. Pertussis, as of 1934, 2,6,500 cases, 269 were there, and as of 2018, only 15,609 cases remained. Polio, as of 1952, there were 21,269 cases, but as we speak of 2018, there was 0 cases. Rubella, as of 1969, there were about 57,686 cases, but in 2018, there were only 4 cases reported. Tetanus, as of 1923, there were about 1,560 cases, but in year 2018, only 23 cases reported. Haemophilus influenza type B, in 1984, there were about 20,000 cases, but in 2018, 38 cases remained. Hepatitis B, as of year 1985, only 26,611 cases were there, but as of now, there were 3,322 cases. This table illustrates the striking decrease in incidence of selected infectious diseases in the United States. Maybe you're in another part of the world, but this is United States. So since the year 1960s, there have been a remarkable transformation in our understanding of immune system and its function. Advances in cell culture technique, including monoclonal antibody production, immunohistochemistry, recombinant DNA methodology, next-generation DNA sequencing, X-ray crystallography, and creation of genetically altered animals, especially transgenic and knockout mice, have changed immunology from a largely descriptive science into one in which diverse immune phenomena can be explained in a structural and biochemical terms. Some of the most important advances in immunology have come since 1990s, with the development of therapies targeting different components of the immune system that are based on fundamental science and are dramatically altering the progression of human inflammatory diseases and cancers. In this chapter, we outline the general feature of immune responses and introduce the concept that form the cornerstone of the modern immunology and that recur throughout this book. Innate and adaptive immunity. Defense against microbes is mediated by sequential and coordinated responses that are called innate and adaptive immunity. And innate immunity, also called natural immunity or native immunity, is essential for defending against microbes in the first few hours or days after infection, before adaptive immune responses have been developed. Innate immune is mediated by mechanisms that are in place even before an infection occurs, hence innate, and are capable of reacting rapidly to invading microbes. In contrast to innate immunity, there are other immune responses that are stimulated by exposure to infectious agent and increase the magnitude and defensive capability with each successive exposure to particular microbes. Because this form of immunity develops a response to infection and thus adapt to infection, it is called adaptive immunity, and also called specific immunity or acquired immunity. The adaptive immunity system recognizes and reacts to a large number of microbial and non-microbial substances called antigen. Although many pathogens have evolved to resist the innate immune response, the stronger and more specialized adaptive immune response are capable of eradicating many of these infections. There are also numerous connections between innate and adaptive immune responses. The innate immune response to microbe provide early danger signal that stimulate adaptive immune response. Conversely, adaptive immune response often work by enhancing the protection mechanism of innate immunity, making them more capable of effectively combating microbes. You will see different diagrams if you follow this book, and here we are explaining some of the features of innate and adaptive immunity. Innate and adaptive immunity. In terms of specificity, innate immunity has molecular shared by groups related to microbes and molecular produced by damaged host cell. For adaptive immunity, for many different microbial and the non-microbial agent. Diversity. Innate immunity has low diversity, recognition of molecules encoded by inherited germline genes. But for adaptive immunity, it has very high. Many antigen receptors are generated by somatic recombinations of gene segment in lymphocyte. In terms of memory, innate has limited memory, while the adaptive immunity has a very great memory. Non-reactivity to self, both of them, they are non-reactive to self. In terms of component, cellular and chemical barrier, the innate has skin, mucosal epithelial, and antimicrobial molecules, while the adaptive immunity has lymphocyte in epithelial, antibodies secreted by epithelial surface as well. Secreted proteins, innate immune has complement and virus lectin, while the adaptive immunity has antibodies. In terms of cells, the innate immunity has a phagocyte, which are macrophage, neutrophil, dendritic cell, natural killer cells, mast cell, innate lymphocyte cells, while the adaptive immune has lymphocytes. Many of these components of innate immunity also serve important function in adaptive immune response, as we will discuss in later chapters. So every individual immune system is able to recognize, respond, and eliminate many foreign non-self antigen, but does not usually react against that individual's own self-antigen on tissue. Different mechanisms are used by innate and adaptive immune system to prevent reaction against health host cells. Mechanisms for defending the host against microbes are present in all multicellular organisms. Phylogenetically, all the mechanisms of the host defense are those of innate immunity, in which are present even in plants and insects. Approximately 500 million years ago, jawless fish such as lampreys and hagfish developed an immune system containing lymphatic-like cells that may function like lymphocytes in more advanced species and even respond to immunization. The antigen receptor of these cells are protein, which are with limited variability that are capable of recognizing many antigens but are distinct from highly variable antibodies and T cell receptor that appeared later in evolution. The more specialized defense mechanisms that constant adaptive immunity are found only in vertebrates. Most of the components for adaptive immune system, including lymphocyte with diverse antigen receptor, antibodies, and specialized lymphoid tissue, evolved coordinatedly within a short time in jawed vertebrates. This is shark approximately 300 years million ago. Innate immunity. The innate immune system responds almost immediately to microbes and injured cells, and repeated exposure induces virtually identical innate immune response. The receptors of innate immunity are specific for structures that are common to group of related microbes and do not distinguish fine difference among microbes. The principal component of innate immunity are physical and chemical barrier, such as epithelial and antimicrobial chemicals produced at epithelial surface, phagocytic cell, for example, neutrophil, macrophage, dendritic cells, mast cell, natural killer cells, and other innate lymphoid cells. Others are blood protein, including component of the complement system and other mediator of inflammation. Many innate immune cells, such as dendritic cells, some macrophage, and mast cell, are tissue resident, and they function as sentinel to keep watch for microbes that may invade the tissue. The innate immune response combats microbes by two strategies: by recruiting phagocytes and other leukocytes that destroy the microbes in the process called inflammation, and by blocking the viral replication or killing virus-infected cell by mechanism distinct from inflammatory reaction. We will discuss the features, mechanism, and component of innate immunity in chapter 4. Adaptive immunity. The adaptive immune response is mediated by a cell called lymphocyte and their product. Lymphocytes express highly diverse receptors that are capable of recognizing a vast number of antigens. There are two major populations of lymphocytes called B lymphocyte and T lymphocytes, which mediate different types of adaptive immune responses. We will firstly summarize the important properties of adaptive immune system and then describe the different types of adaptive immune responses. The cardinal features of adaptive immune responses. The fundamental properties of the adaptive immune system reflect the properties of lymphocytes that mediate these responses. First, specificity and diversity. Immune responses are specific for distinct antigen and are often for different portions of a single complex protein, polysaccharide, and other macromolecules. The part of complex antigen that are specifically recognized by lymphocytes are called determinants or epitopes. This fine specificity exists because individual lymphocytes express membrane receptors that are distinguish substructure difference between distinct epitopes. Clones of lymphocytes with different specificity are present in an immunized individual and are able to recognize and respond to foreign antigen. This fundamental concept is called clonal selection. It was clearly enunciated by Macrophel Barnett in 1957 as a hypothesis to explain how the immune system could respond to large number and variety of antigen. According to this hypothesis, which is now proven feature of adaptive immunity, antigen-specific clones of lymphocytes develop before and independent of exposure to antigen, and introduce the antigen bind to select the cells of pre-existing antigen-specific clone and activate them, leading to immune response specific for that antigen. The total number of antigen specificity of the lymphocyte in an individual called the lymphocyte repertoire is extremely large. It is estimated that immune system of an individual can distinguish 10 power 7 to 10 power 9 distinct antigen determinants. This ability of lymphocyte repertoire to recognize a very large number of antigen called diversity is the result of variability in structure of antigen binding sites of lymphocyte receptor for antigen. In other words, there are about many different clones of lymphocyte, and each clone has unique antigen receptor and therefore a singular antigen specificity contributing to total repertoire that is extremely diverse. The expression of different antigen receptor in different clones of T and B cell is a reason why the receptor are said to be clonally distributed. The molecular mechanism that generates such diversity antigen receptor are discussed in chapter 8. Diversity is essential for immune system. Diversity is essential if the immune system is to defend the individual against many potential pathogens in the environment. Memory. Exposure of immune system to foreign antigen enhance its ability to respond again to the antigen. Response to the second and subsequent exposure to the same antigen called secondary immune response are usually more rapid, greater in magnitude, and often qualitatively different from the first or primary immune response that antigen to respond to that antigen. Immunological memory occurs because each exposure to an antigen generates long-lived memory cells specific for antigen. There are two reasons why secondary response are typically stronger than primary immune response. Memory cells accumulate and become more numerous than the naive lymphocyte specific for antigen that exist at time of initial antigen exposure, and the memory cell react more rapidly and vigorously to antigen challenge than do naive lymphocytes. Memory enables the immune system to mount heightened response to persistence or recurring exposure to the same antigen and thus combat infection by microbes that are prevalent in environment and encountered repeatedly. Non-reactivity to self or self-tolerance. One of the most remarkable properties of every normal individual's immune system is its ability to recognize, respond to, and eliminate many foreign non-self antigen while not reacting to harmful to that individual's own cells or antigen. Immunological unresponsiveness is also called the tolerance. Tolerance to self-antigen or self-tolerance is maintained by several mechanisms. These include eliminating lymphocytes that express receptor specific for some self-antigen, inactivating self-reactive lymphocyte, or suppressing these cells by action of other regulatory cells. Abnormality in induction or maintenance of self-tolerance lead to the immune response against self, autologous antigen, which may result in disorder called autoimmune disease. The mechanism of self-tolerance and its failure are discussed in chapter 15. In addition to these cardinal features of adaptive immunity, these responses have some other important properties. Because of ability of lymphocyte and other immune cells to circulate among tissues, adaptive immune system is systemic, meaning that even if an immune response is initiated at one site, it can provide protection at distant site. This feature is, of course, essential for success of vaccination. A vaccine administered in the subcutaneous or muscle tissue of the arm can protect from infections in any tissue. Immune responses are regulated by a system of positive feedback loop that amplify the reaction and by control mechanisms that prevent inappropriate or pathological reaction. When lymphocytes are activated, they trigger the mechanism that further increase the magnitude of response. This positive feedback is important to enable a small number of lymphocytes that are specific for any microbe to generate the large response needed to eradicate the infection. Many control mechanisms become active during immune response, which prevent excessive activation of lymphocytes that could cause a collateral damage to tissue and also to prevent a response against self-antigen. An overview of humoral and cell-mediated immunity. There are two types of adaptive immunity called humoral immunity and cell-mediated immunity, which are mediated by different types of lymphocytes and function to eliminate different types of microbes. Humoral immunity is mediated by a molecule in the blood and mucosa secretion called antibodies, which are produced by B lymphocytes. Antibody recognize microbial antigens, neutralize the infectivity of microbes, and target microbes for elimination by phagocytes and complement system. Humoral immunity is the principal defense mechanism against microbes and their toxin located outside the cell. This is in the lumen of gastrointestinal or respiratory tract in the blood. Because secreted antibody can bind to these microbes and toxin, neutralize them, and assist in their elimination. Cell-mediated immunity, also called cellular immunity, is mediated by T lymphocyte. Many microbes are ingested but survive within phagocyte, and some microbes, notably viruses, infect and replicate in various host cells. In this location, the microbes are inaccessible to circulating antibodies. Defense against such infections is a function of cell-mediated immunity, which promotes the destruction of microbes inside the phagocyte and the killing of infected cells to eliminate reservoir of infection. Different classes of lymphocytes may be distinguished by expression of membrane protein, many of which are designed by CD number. These surface molecules are also involved in the function of lymphocyte. We will introduce some of the surface molecules that are used to identify lymphocyte classes in Chapter 2 and discuss them further later in chapters. A summary of molecular designed by CD number mentioned in the book is provided in Appendix 1. You can read yourself. Protective immunity against a microbe may be provided either by host's response to the microbes or by transfer of antibody that defend against the microbes. The form of immunity that is included by exposure, that is induced by exposure to foreign antigen, is called active immunity, because the immunized individual play an active role in responding to an antigen. Individual and lymphocytes that have not encountered a particular antigen are said to be naive, implying that they are immunologically inexperienced. Individuals who have responded to microbes, antigen, and or are protected from subsequent exposure to that microbes are said to be immune. Immunity also can be conferred as an individual by transferring antibody from an immunized individual into an individual who has not encountered the antigen. The recipient of such transfer becomes immune to the particular antigen without having been exposed to or having responded to the antigen. Therefore, this form of immunity is called passive immunity. A physiologically important example of passive immunity is the transfer of maternal antibodies through the placenta to the fetus, which enables a newborn to combat infection for several months before they develop the ability to produce antibody themselves. Passive immunization is also a medically useful method for conferring resistance rapidly without having to wait for an active immune response to develop. Passive immunization against the potentially lethal toxin by the administration of antibody from immunized animals or people is a life-saving treatment for rabies infection and snake bites. Patients with some genetic immunodeficient diseases are passively immunized by transfer of pooled antibody from health donors. The first demonstration of humoral immunity was provided by Emil von Belhem and Shiba Sarobin Kitasato in 1890s using a passive immunization strategy. They showed that if serum from animal that has been immunized with attenuated form of diphtheria toxin was transferred to a naive animal, the recipient became specifically resistant to diphtheria infection. The active component of the serum was called antitoxin because they neutralized the pathological effect of diphtheria toxin. These results led to the treatment of otherwise lethal diphtheria infection by the administration of antitoxin, an achievement that was recognized by a award of the first Nobel Prize in Physiology or Medicine to von Behring. In the 1890s, Paul Ehrlich postulated that immune cells use receptors which are called the side chain to recognize microbial toxin and subsequently secrete these receptors to combat microbes. He coined the term antibodies, Antikörper in Germany, for the serum protein that bound for foreign substances such as toxin, and the substance that generated antibody were called antigen. The modern definition of antigen include molecules that bind to specific lymphocyte receptor, whether or not they stimulate immune responses. According to strict definition, substances that stimulate immune response are called immunogens, but the term antigen is often used interchangeably with immunogen. The properties of antibodies and antigen are described in chapter 5. Ehrlich's concept were the remarkably present model of the specificity of adaptive immunity. These early studies of antibody led to the general acceptance of humoral theory of immunity, according to which host defense against infections is mediated by substance present in the body fluid, once called humors. Ila Metchnikoff initially championed the cellular theory of immunity, which stated that host cells are the principal mediator of immunity. His demonstration of phagocytes surrounding the thorn stuck into the translucent starfish larva, published in 1883, was perhaps the first experimental evidence that cells respond to foreign invaders. Ehrlich and Metchnikoff shared the Nobel Prize in 1908 in the recognition of their contribution to establish this fundamental principle of immunity. Sir Almroth Wright observation in the early 90s that factor in immune serum enhance the phagocytosis of bacteria by coating the bacteria, process known as opsonization, lent support to the belief that antibodies prepare microbes for ingestion by phagocytosis. These early cellulists were unable to prove that specific immunity to microbe could be mediated by cell. The importance of cellular immunity in host defense became firmly established in the 1950s when it was shown that resistance to intracellular bacteria, Listeria monocytogenes, could be transferred to the animal with cell but not with serum. We now know that specificity of cell-mediated immunity is due to T lymphocyte, which often function in concert with other cells such as phagocyte to eliminate microbes. In clinical setting, immunity to previous encountered microbes is measured indirectly, either by assaying for the presence of product of immune response, such as serum antibody specific for microbial antigen, or by administration of substance purified from microbes and measuring reaction to this substance. A reaction to an antigen is detectable only in individuals who are previously encountered the antigen, reflecting memory of that antigen. These individuals are said to be sensitized to an antigen. and the reaction is an indication of sensitivity. Such a reaction to microbial antigen implies that the sensitized individual is capable of mounting a protective immune response to microbes. Initiation and development of adaptive immune response. An adaptive immune response develops in several steps, starting with the capture of antigen followed by activation of specific lymphocytes. Most microbes and other antigen enter through epithelial barrier and colonize the tissue, and adaptive immune response to these antigen develop in secondary peripheral lymphoid organs. The initiation of adaptive immune responses requires that antigen be captured and displayed to specific lymphocyte. The cells that serve this role are called antigen-presenting cell, APC. The most specialized antigen-presenting cell are dendritic cell, DC, which capture microbial antigen that enter from the external environment, transport these antigen to lymphoid organ, and present the antigen to naive T lymphocyte to initiate immune response. Other cell types function as antigen-presenting cell at different stages of cell-mediated and humoral immune response. We will describe the function of antigen-presenting cell in chapter 6. Naive lymphocytes express antigen receptor but have not responded to antigen. The activation of these lymphocytes by antigen leads to the proliferation of these cells, resulting in an increase in the size of antigen-specific clone called clonal expansion. This is followed by differentiation of activated lymphocyte into cell capable of eliminating antigen called effector cell because they mediate the ultimate effect of immune responses, and memory cell that survive for long period and mount strong response to repeat antigen encounter. Antigen elimination often require the participation of others, non-lymphoid cells such as macrophage, neutrophil, which are also sometimes called effector cells. These steps in lymphocyte activation and differentiation into effector cell typically take few days, which explain why adaptive immune response is slow to develop and innate immune has to provide protection initially. After the adaptive immune response has eradicated the infection, the stimuli of lymphocyte activation dissipate and most of the effector cells die, resulting in the decline of response. Memory cells remain ready to respond vigorously if the same infection recurs. The cells of the immune system interact with one another and with other host cells via secreted protein called cytokines. Such interactions are essential during both initiation and effector stages of innate and adaptive immune responses. Cytokines are a large group of secreted proteins with diverse structures and function, which regulates and coordinates many structures and function, which regulate and coordinate many activities of the cell of innate and adaptive immunity. All cells of immune system secrete at least some cytokines and express specific signaling receptor for several cytokines. Among the many functions of cytokine we will discuss through this book are promoting the growth and differentiation of immune cell, activating the function of lymphocyte and phagocytic that eliminate microbes called effector function, and stimulating directing movement of immune cells from blood into tissue and within tissue. A large subset of structure-related cytokines that regulate cell adhesion and migration are called chemokines. Cytokines are also involved in immunological diseases, and some of the most effective drugs developed to treat these diseases target cytokines. We will describe the function of individual cytokines when we discuss immune response, in which these proteins play important roles. A list of cytokines and brief summary of their properties are provided in an appendix 2. Humoral immunity. B lymphocytes that recognize antigen proliferate and differentiate into plasma cells that secrete different classes of antibodies with distinct functions. Each clone of B cell expresses a cell surface antigen receptor, which is a membrane bound from antibodies with a unique antigenic specificity. Many different types of antigen, including proteins, polysaccharide, lipid, and small molecules, are capable of eliciting antibody responses. The response of B cell to protein antigens require activating signal help from CD4 plus T cell, which is the historical reason for calling this T cell helper cell. B cell can respond to many non-protein antigen without participation of helper T cell. Each plasma cell secretes antibodies that have the same antigenic binding site as the B cell surface antigen receptor that first recognize the antigen. Polysaccharide and lipid stimulates secretion mainly of the antibody class isotope called immunoglobulin M, IgM. Protein antigen induce the production of antibodies of different classes, IgG, IgA, and IgE, from a single clone of B cell, a process called heavy chain class or isotope switching. These different antibody classes serve distinct function mentioned later. Helper T cell also stimulate the production of antibodies with increased affinity for the antigen. This process, called the affinity maturation, improve quality of humoral immune response. The humoral immune response combats microbes in many ways. Antibody bind to microbes and prevent them from infecting the cell, thus neutralizing the microbes. Antibody-mediated neutralization is the only mechanism of adaptive immunity that stops the infection before it is established. This is why eliciting the production of potent neutralizing antibody is the key goal of vaccination. IgG antibodies coat microbes and target them for phagocytosis because phagocyte, neutrophil, and macrophage expresses receptor for parts of IgG molecule. IgG and IgM activate the complement system, and complement products promote phagocytosis and destruction of microbes. IgA is secreted from mucosal epithelium and neutralizes microbes in lumens of mucosal tissue such as the respiratory and gastrointestinal tract, thus preventing inhaled and ingested microbes from infecting the host. Maternal IgG is actively transported across placenta and protect the newborns until baby's immune system become mature. Most of IgG antibodies have half-lives in the circulation of approximately 3 weeks, whereas the other classes of antibody have half-lives of just few days. Some antibodies secreting plasma cells migrate to the bone marrow and mucosal tissue and live for years, continuing to produce low level of antibodies. The antibodies that are secreted by these long-lived plasma cells provide immediate protection if the microbes return to infect the individual. The most effective protection is provided by memory cells that are activated by microbes and rapidly differentiate to generate a large number of plasma cells. Cell-mediated immunity. T lymphocyte, the cell of the cell-mediated immunity, recognize antigens of self-associated microbes and different types of T cell help phagocytes to destroy these microbes or kill the infected cell. T cells do not produce antibody molecules. Their antigen receptor are membrane molecules distinct from but structurally related to antibodies. See Chapter 7. T lymphocytes have a restricted specificity for antigen. They recognize peptides derived from foreign protein that are bound to host protein called major histocompatibility complex molecule, which are expressed on the surface of other cells. As a result, these T cells recognize and respond to cell-associated but not soluble antigens. T cell consists of functionally distinct populations, the best defined of which are helper T cell and cytotoxic or cytolytic T lymphocyte, CTLs. Helper T cell function mainly by secreting cytokine and membrane molecule which activate other cells to kill microbes, whereby cytotoxic cell produce a molecule that directly kill infected cell. Some T lymphocytes, which are called regulatory T cells, function mainly to inhibit immune response. We will return to more detailed discussion of the properties of lymphocytes in chapter 2 and later in chapters. Upon activation in a secondary lymphoid organ, naive T lymphocytes differentiate into effector cell, and many of them leave the lymphoid organ and migrate to the site of infection. When these effector T cells again encounter cell-mediated microbes, they are activated to perform the functions that are responsible for eliminating the microbes. Cytokine produced by CD4 plus helper T cell recruit leukocyte, and both cytokines and plasma membrane protein stimulate production of microbial substance in phagocyte. Thus, these T cells help phagocytosis to kill the infectious agents or pathogen. Other CD4 helper T cells secrete cytokines that help B cell to produce type of antibody called IgE and activate leukocyte called eosinophils, which are able to kill helminths that may be too large to phagocytosis. Some CD4 T cell helper T cell stay in lymphoid organ and use membrane molecule and cytokine to stimulate B cell to make highly effective and functional specialized antibodies. CD8 cytotoxic T cell kill cell harboring microbes in cytoplasm. These microbes may be virus that infect many cell types or bacteria that are ingested by macrophage but escape from phagocytic vesicles into cytoplasm, where they are inaccessible to the killing machinery of phagocyte, which is largely confined in vesicle. By destroying the infected cell, cytotoxic T cell eliminate the reservoirs of infection. Cytotoxic T cell also kill tumor cells that express the antigen that are recognized as foreign. In the remainder of the book, we describe the detail of recognition, activation, regulation, and effector phases of innate and adaptive immune response. The principles introduced in this chapter recur throughout this book. Summary of the chapter. Protective immune against microbe is mediated by early reactions of innate immunity and later response of adaptive immunity. Innate immune response are stimulated by molecule structure shared by a group of microbes and by molecule expressed by them, the host cell. Adaptive immunity is specific for different microbial and non-microbial antigens, and is increased by a repeated exposure to antigen, immunological memory. Many features of adaptive immunity are of fundamental importance for its normal function. These include specificity for different antigen, a diverse repertoire capable of recognizing a wide variety of antigen, memory of antigen exposure, and ability to discriminate between foreign antigen and self-antigen. Immunity may be acquired by a response to antigen, active immunity, or conferred by transfer of antibody or effector cell, that is called passive immunity. Lymphocytes are the only cells capable of specifically recognizing antigen, and thus the principal cells of adaptive immunity. The total population of lymphocytes consists of many clones, each with unique antigen receptor and specificity. The two major subsets of lymphocytes are B cell and T cell, and differ in their antigen receptor and function. The adaptive immune response is initiated by recognition of foreign antigen by a specific lymphocyte. Specialized antigen presenting cell capture microbial antigen and display these antigen for recognition of lymphocyte. Lymphocytes respond by proliferation and differentiating into effector cell. Whose function is to eliminate antigen and into memory cell, which shows enhanced response on subsequent encounter with antigen. The elimination of antigen often require the participation of various effector cell. Humoral immunity is mediated by antibody secreted by B cell lymphocyte and their differentiated progenies, plasma cell, and is the mechanism of defense against extracellular microbes. Antibody neutralize the infectivity of microbes and promote the elimination of microbes by phagocytes and by activation of the complement system. Cell-mediated immunity is mediated by T lymphocyte and their product, such as cytokine, and it is important for defense against intracellular microbes. CD4 helper T lymphocyte help phagocytes to eliminate ingested microbes and help B cell produce antibodies. CD8 cytotoxic T lymphocytes kill cells harboring intracellular pathogen, thus eliminating reservoir of infection. The many selected readings, you can go and have a look. Otherwise, this was the end of chapter 1. Congratulations for being together with me throughout this session. Amen.